Good morning. Now we are starting a new section, and we have a plenary conference. This conference will be performed by Dr. Ahmad Saad from Strasbourg University and Professor Jarbas Magalhães will be the chair of this section. Please.
So thank you, Mariana. It's my utmost pleasure to introduce Dr. Ahmad Saad. Ahmad earned his master's degree in biochemistry from the University of Bordeaux in France, the city that he's chosen to stay for his PhD studies under the supervision of Professor Eric Dufoc. Living in Bordeaux, he was actually working with wines and vines, and he studies the influence of lipids on food matrices and how they interfere in the organoleptic properties when tasted together with wine. So something really related to the city. And he has specialized in solid-state NMR spectroscopy during his PhD studies. And then he moved out to Strasbourg, where he's joined the team of Professor Burkhard Bechinger, where he works with peptide membrane interactions, developing several biophysical approaches, especially related to solid-state NMR spectroscopy to study peptide membrane interactions, and he has a major focus on multi-resistant bacteria. So when we think of NMR, you can think it's a bit tricky. If you think of solid-state NMR spectroscopy, it's definitely trickier. But the huge advantage of solid-state NMR then is that you can explore larger systems like this more realistic protein-peptide membrane interactions. So thank you so much, Ahmad.
Thank you, Jarbas, for the kind introduction. So thank you all for being here today. I am a postdoctoral researcher working at the Membrane Biophysics and NMR team under supervision of Professor Burkhard Bechinger. And the thematic of my talk today is about antimicrobial resistance and how we use biophysical tools to investigate antimicrobial peptides and membrane environment in order to understand their mechanism of action. So first, since the discovery, nowadays there is like a silent pandemic spreading worldwide represented by the bacterial resistance to antibiotic. Since the discovery of antibiotic, bacteria are capable to develop resistance after a while. And this resistance is due to several factors such as natural resistance or mutation, genetic mutation, or the overuse of antibiotic. And according to World Health Organization, this resistance could lead to a huge amount of deaths in 2050. And this represents a serious threat for human health, development, and even economy today. This is why an alarm is going on until the development of new tools to overcome this resistance. And one of the approaches is the uses of antimicrobial peptides. So antimicrobial peptides are a small sequence of amino acid. We can naturally, they are naturally present in several types of species like mammalian, amphibian, plants, or insects. They have a potential therapeutic class. They are potential therapeutic class of antimicrobial, and they are known to interfere with the barrier function of the bacterial membrane and cause bacterial killing, and this provide them resistance, a robust resistance against bacteria. From a structural point of view, we can find them as beta sheet structure, alpha helical, or random coil. They are not only antimicrobial. We saw in the morning some example of other biological activity, like in the case of Alzheimer's disease or antifungal, anti-inflammatory, and anti-cancer. And several peptides has been developed, and as we can see in this table, some are in a clinical trial stage. But before reaching this stage, we need to understand how the peptide behave. We need to understand their biological activity. Are they active against what species of bacteria? Are they cytotoxic? We need also to understand their biophysical activity, their structure, how they are structured, what are their site of interaction, like lipids, lipopolysaccharide, the cell wall of bacteria, in order to understand their mechanism of action. And if we look to the literature, we can find such different type of mechanism of action of antimicrobial peptides, such as the toroidal pore model. When the peptide partition into the lipid bilayer, they can form these transmembrane helix and form pore into the bacterial membrane. Another model represented by the carpet model, where they still at the membrane interface, they form this carpet mode distribution. Another mechanism of action proposed by Professor Burkhard Baschinger, represented by the SMART model, soft membranes adapt and respond transiently. Indeed, it's an interplay between peptide and lipid, and depending on factors such as peptide concentration, lipid composition, fluidity of the lipid bilayer, or charges, we can have several types of peptide-lipid interaction leading to the disintegration of the lipid bilayer and causing the bacterial killing. In our team, we are interested with peptide of the magainin family, and magainin 2 and PGLA are two peptides naturally present in the skin of the Xenopus laevis frog. They are two cationic amphipathic peptides. As we can see, we have several positively charged lysine in the amino acid sequence of MAC2 or PGLA, and the amphipathic character represented by the site where we have all the hydrophilic residues and the other side of the hydrophobic residues. These peptides adopt a random coil structure in solution, but when we add lipids, they adopt an alpha helical structure. As we can see here, the structure of MAC2 in DPC micelles. From a biological point of view, these peptides are effective against bacteria. We have calculated their biological activity. It's around the micromolar range. And as we can see on this SEM image, in the presence, for example, of MAC2, we have like this deformation in the membrane of the E. coli cells, showing that these peptides interfere with the membrane of the bacteria. But what is interesting about these peptides is when they are mixed together at 1:1 ratio, they show enhanced antimicrobial activity. So they work in a synergistic mode of action. And this synergism is not present only on biological sample like E. coli bacteria, but it's also on membrane model. They showed some lytic activity and pore formation, and they work also synergistically. We have also demonstrated that in the presence, for example, for MAC2, the binding of them to the lipid bilayer to the membrane is enhanced in the presence of the other peptide, PGLA. And we have also shown that these peptides are capable to form some supramolecular distribution at the lipid membrane. And we are interested to understand this synergistic interaction between these two peptides. And our hypothesis is that the peptides partition into the lipid bilayer, they start to interact together. And at high peptide to lipid density, these peptides could create pores leading to the leakage and the destruction of the kill of the bacteria. So this is why we will use some high resolution investigation at atomic level to investigate peptide-lipid assemblies and peptide-peptide interaction in order to understand this synergism. And this is done in a membrane environment. And if we go a bit into biology, a lipid bilayer, a cell membrane is represented by the fluid mosaic model. It is a large assembly of proteins, carbohydrates, and lipid. It's characterized by a fluid behavior, and it serves of different functions such as protection, transportation through some protein channel, or communication between the intra and the extracellular cell. And if we want to study membrane proteins or peptide membrane interaction, it's important to respect the physical behavior of the lipid bilayer, the lipid composition, the curvature, and the charges. And if we look to the literature, if we want to study interaction with membrane, we can find different type of membrane mimetic systems, such as micelles. These are small molecule of detergent. Bicelles, which are also small molecule of lipid bilayer represented by like a disc, composed of long chain and short chain lipids. And we have liposomes. And to get atomic level information about peptide lipid assembly, the only existing technique that could provide us this structural information is solid-state NMR, where we can use liposome system. The advantage of using liposome systems that it's a bilayer structure. We can tune liposome with different type of lipid, like charged lipid, cholesterol. So we are very close to the native-like environment of the cell membrane. So to do so, we use solid-state NMR. We have different existent approaches of solid-state NMR, such as magic angle spinning. The idea is to prepare vesicles. We put them inside this small spinner, and we spin the sample at high frequency. Why we do this? Multilamellar vesicles are a large assembly of lipids, and they move very slowly. Due to this slow tumbling, we will have an anisotropic behavior that will lead to a very low resolved spectra in NMR. So we spin the sample at this angle where the anisotropic interaction are averaged out, so we can get this highly resolved spectra. We can assign it and get some structural information. And we can go in spinning from 15 kilohertz to 100,000 kilohertz, depending on the rotor side. Another approach, which is also developed by our team, represented by oriented solid-state NMR, oriented membrane solid-state NMR. So we prepare lipid bilayer on a glass plate, and these glass plates are oriented with the magnetic field. And the chemical shift of the nitrogen-15 is indicator of the helix orientation with the magnetic field. So when the helix is oriented parallel to the magnetic field, we have an in-plane topology of the peptide, meaning the peptide is at the surface parallel to the membrane surface. While when we have a chemical shift above 100 ppm to 100 ppm, we have a transmembrane helix. And through this approach, we can see how the peptide partition into the lipid bilayer. Other solid-state NMR approaches that we can use also to study peptide membrane interaction. Here it's more focused on lipids by using deuterium solid-state NMR to get information about the dynamic of the lipids, how the lipid acyl chain move, or even the morphology of the lipid bilayer thanks to the phosphorus NMR. And in the next slide, I will show you some example how we use this technique to get information about this peptide-lipid interaction. So first of all, we have investigated the topology of magainin 2 and PGLA in different membrane with different lipid composition. Here we have DMPC, DMPE membrane. These are lipids with saturated acyl chain. While POPC or POPE, these are lipids with unsaturated acyl chain. And in the nature, the lipid, the cell membrane of bacteria or of mammalian cells are more rich in unsaturated lipids. So we have investigated the topology. For magainin 2, for example, whatever is the lipid composition, the peptide is always in an in-plane topology. While for PGLA, we have found that the topology of PGLA is depending on the lipid composition of the membrane. In DMPC or DMPE membrane, PGLA adopt a transmembrane orientation, as we can clearly see here through the chemical shift of the N15 signal. While in POPC or POPE, POPG membrane, the peptide adopt a more an in-plane topology, thanks to the N15 chemical shift, indicating that PGLA alignment is lipid dependent. And if we do the comparison between the different lipid composition, for example, PC lipids have the volume of the polar head is higher than the PE lipids. So the PE creates some negative curvature to the lipid bilayer. So we clearly see that the topology of the peptide is not really related to the curvature of the lipid bilayer, but it's more driven by the lipid acyl chain. On the other hand, we have investigated the impact of the lipid on the membrane dynamic thanks to deuterium solid state NMR. So before presenting the data, we use some deuterated chain lipids. In NMR, deuterium is a spin 1 nuclei, so the NMR signal is called quadripolar splitting. The signal for one CD bond is represented by a doublet, the peak doublet. The difference between the two peaks is called the quadripolar splitting. And this quadripolar splitting is depending on the dynamic of the CD bond. When the dynamic increase, the splitting decrease, and vice versa. So in the case of a lipid bilayer composed of different, where we have different CD bond, the NMR signal is represented by overlap of doublet corresponding to each position on the lipid acyl chain, while the doublets with the lowest splitting here correspond to the terminal methyl. It's a very dynamic methyl, and the other position correspond to the other position on the lipid acyl chain. And from these type of spectra, we can extract two types of information about the chain order parameter. When we have an order parameter close to 1, this means that we have an ordered lipid bilayer. There is no movement. And when it decreases, this means that we have an increase in the fluidity of the lipid bilayer. On the other hand, we can also extract some information related to the gel to fluid transition of the lipid bilayer thanks to first spectral moment by acquiring NMR spectra at different temperature. So we can also investigate the physical state of the lipid membrane. So in the present slide, we have several deuterium NMR spectra of a POPE, POPG membrane. Why we use these lipids? Because these are the most abundant lipids on a Nicolai cell membrane. And here, POPE is deuterated. And we can clearly see this gel to fluid transition through the just difference in shape in the deuterium solid state NMR spectra. At low temperature, we have a gel phase. We don't have any resolution. We can detect these quadripolar splitting. So we acquired the experiment in presence and in the absence of the peptide. As we can see here, in presence of MAC2, we have, or PGLA, we have decrease in the M1. The effect is more pronounced at high temperature in a fluid membrane compared to a gel phase membrane, meaning that the physical state of the membrane could have an impact on the peptide-lipid interaction. On the other hand, in the presence of, if we extract the chain order parameter, we can clearly see a decrease in the order parameter of the lipid acyl chain, indicating that the peptides are disordering the lipids, the lipids of the membrane. We have done this experiment with different lipid composition, PCPG membrane that mimic more like a mammalian cell membrane, PEPG to mimic bacteria, and this is the relative order parameter. And we have labeled or the Zwitter ionic lipid or the negatively charged lipid represented by PG. And if we compare between a mammalian membrane and a bacterial membrane, we can clearly see that the effect is more pronounced on a bacterial membrane. And if we compare also between a Zwitter ionic membrane and a negatively charged membrane, we can clearly see also that the disordering effect is more important on a negatively charged membrane. And this is logic since we are dealing with cationic peptides. How to explain this disordering effect of the peptide on the lipid acyl chain is more also related to the topology. When they are inserted at the polar head of the lipid, they can insert between the polar head, causing their expansion. That creates some gaps at the core of the lipid bilayer, meaning that the lipid acyl chain are becoming more dynamic. Then we wanted to investigate to get some structural information about the peptide, how they are folded in multilamellar vesicles, and if we can detect also some peptide-peptide interaction in the context of the synergy. This is why we have synthesized the peptide with some selectively labeling. We have labeled this green position on each amino acid sequence of the peptide with T13 and N15. And we have chosen this position because according to literature, when they do some mutation of some of these residue, the synergy between the two peptides is lost. So after the synthesis of the peptide, we have reconstituted them in the multilamellar vesicle, and we have acquired magic angle spinning solid state NMR. And in such type of a technique, we use two types of reference experiment. The first one we called INAPT. In INAPT, we have like a polarization transfer from a proton to a carbon through the bonds. So we can detect everything flexible. While CP, which is a cross-polarization, the transfer from a proton to a carbon is through dipolar interaction through space, we detect everything rigid. So this is an example of a carbon spectra of a lipid membrane composed of POPE, and we clearly see that we have a very weak CP signal compared to the INAPT due to the dynamic behavior of the lipid bilayer. We have applied this also to magainin 2, reconstituted in this type of lipid bilayer. This is the carbon-13 spectra of, which is considered as a fingerprint of the peptide. In this region, we can distinguish the aliphatic amino acid, the alpha, the aromatics, and the carbonyl. And if we look into the details of these spectra, we can clearly see that the peptide signals are present in the CP experiment. But in the INAPT, we still have also some resonances corresponding to the peptide. And interestingly, we can clearly see, for example, the beta ring of the phenylalanine is absent in the INAPT, but present in the CP, while for the lysine, the epsilon carbon of the lysine is absent in the CP and present in the INAPT, meaning that some residues of the peptide are having some isotropic motion in the lipid membrane. And we can explain this also through the partitioning of the peptide into the lipid bilayer. For example, phenylalanine is like more hydrophobic residue, is more inserted into the lipid bilayer, while lysine is outside of the lipid bilayer and having more dynamic behavior. Then we wanted to know more about the folding. What is the structure of the peptide in liposomes of PEPG? So we acquired also more solid-state NMR spectra. This is a DAR experiment. It's a carbon-carbon correlation experiment where we do some polarization transfer from proton to carbon, then from carbon to another carbon which is closed in space. And this allows us to assign all the carbons for one spin system. And if we increase the mixing time, we can also have some inter-residual correlation. And through this experiment, we can assign the different carbon on the 2D spectra to calculate the secondary structure. This is the result related of magainin 2. The resolution is not really high because of the dynamic behavior of the peptide, but we were capable to assign some residue to see some correlation, for example, between C alpha of the valine, C beta, C gamma, C delta. We didn't observe any molecular correlation, but by taking the chemical shift values, we were able to calculate the secondary structure of the peptide. And as we can clearly see, according to the Ramachandran plot, it's an alpha helical conformation, which is also consistent with liquid state NMR data in DPC mice cells. Interestingly, also we have, if we do some zoom in on some of the peak, we see that for the same residue, for example, for the valine, we are observing two splittings, meaning that when the peptide is partitioning to the lipid bilayer, it might have some heterogeneous structure. We have two populations of peptide, and this might be also explained that maybe that we have a population of P peptide and another peptide which is oligomerized with another one. On the other hand, we have also investigated the topology of this peptide in PE-PG membrane. This is a phosphorus NMR spectra. The signal here means that the peptide is aligned with the magnetic field. On the other hand, the small signal here is of unaligned lipids, and this is, in fact, the effect of the peptide. If it is disordering or impacting the dynamic of the lipids, some lipid will be aligned, some other no. So this explains this signal. While here the N15 NMR signal is between 100 and 135 ppm, which is more obvious with an in-plane topology with some also heterogeneous partitioning due to the contribution around 135 ppm. And this could be explained also due to the peptide to lipid ratio at 4%. Maybe not all the peptides are well distributed on the lipid bilayer, and some of them are not well aligned. On the other hand, we have done the same experiment with PGLA, and we have also calculated the secondary structure according to the chemical shift. The peptide is also alpha helical, while the lysine are not really structured. And during the acquisition of this experiment, we had find out that the sample is undergoing some structural rearrangement over time. We saw some changes in the NMR spectrum. As we can clearly see on this 1D carbon NMR spectrum, day 1, day 2, day 3, the signal here are becoming more intense. Some changes are observed here. Then we continue to acquire data on this sample, and we had a very nice 2D experiment. We assigned a different spin system. We saw also some intra and intermolecular cross-peak, meaning that the peptide is really ordered in the lipid bilayer. But surprisingly, when we did the chemical shift calculation for the secondary structure, we find out that PGLA is adopting a beta sheet structure, which is really inconsistent with the previous data. We took out the sample from the rotor. On this 2D map, we can clearly see the transition, the shift of the peak. We took out the sample of the rotor, and we performed some TEM experiment, and we clearly see some oligomerization of the peptide with lipid. This trend was reproducible through different samples for PGLA, and also for magainin 2, meaning that there is something is happening, meaning that the peptides are capable to form these oligomeric structure with time with lipids. And we have looked also to the lipids. We were wondering what's happening, so we decided also to look to the lipid signal. And we have also saw that with time, some structural changes also is happening with the lipids, meaning that also the driving force of this structural rearrangement is through the lipids. So from the first part that I presented here, we can clearly see that the physical state of the lipid bilayer influences the peptide lipid interaction and folding, like factors such as hydration, temperature are important to be considered. It's also possible to get high-resolution NMR data in realistic membrane that mimic an E. coli bacteria, effectively using the inherent scale properties of the lipid bilayer, such as fluidity or curvature. From a secondary structure point of view, the peptides are in alpha helical conformation. On the other hand, we show that they can also form some supramolecular structure characterized by beta sheet with lipid bilayer. And in a previous study in our team, we have shown that PGLA, magainin 2 are capable also to form some fibrillar structure only in phosphate environment. And I think it was well presented, these THT curves in the previous talk. When we have a cross beta sheet structure, THT is a very nice probe. And PGLA, for example, was showing this behavior. And the lipids are phospholipids, so we have also phosphate group in lipids. So I was wondering if lipids are also triggering this peptide assembly. So we performed some THT experiment of PGLA in absence and in presence of lipids, and we can clearly see that we have a THT signal, meaning that also lipids are the driving force of this peptide assembly. On the other hand, we were interested to see peptide-peptide interaction. So the idea was to mix the two peptides together, to labeling, and trying to see if we have any cross-peak between the peptides. Unfortunately, the chemical shift values are very close between magainin-2 and PGLA. But interestingly, we observed that for magainin-2, we started to see some intermolecular correlation, meaning that the peptide is becoming more ordered in the presence of the other peptide. On the other hand, if we look to the inept spectrum here of magainin-2 alone, of magainin-2 plus PGLA, we can see also that the signals that they were present in, for the peptide alone are vanished in presence of other peptide, meaning that magainin-2 is also becoming more rigid or more ordered in the presence of PGLA. We have also tried to see if we can have any information through variation of chemical shift, because chemical shift is sensitive to the environment, electronic environment around each atom. And we have performed some 2D experiment of PGLA, labeled PGLA alone, and labeled PGLA in presence of unlabeled magainin-2. And we observe also that we have some shift in some peaks, especially here for the alanine residues, meaning that there are some interaction between these two peptides. Another ongoing work on this project is represented to get the full structure of the peptide. This is why to label the peptide, it's very expensive through chemical synthesis. We use bacterial overexpression to produce the peptide. Since these are antimicrobial peptides, so we don't want them to kill the bacteria during the production process, so we produce them with a fusion protein represented by TAF12 with a cleavage site between glutamic acid and proline. We add some carbon-13 labeled glucose or nitrogen-15 ammonium chloride to label the peptide. We have succeeded to produce the peptide and to purify it, and we are starting to get some, to collect some structural data on this peptide in PEPG membrane. But one of the advantages of using this approach is that we are capable to produce the peptide or only C13 labeled or only N15 labeled. And the idea here is to mix the two peptides with two different labels and try to do some NC transfer. So if we have this NC transfer, this means that the peptides are close to each other. And in solid-state NMR, we have an experiment called REDOR. And REDOR is an experiment that we use to calculate the distance between two atoms. And the experiment is simple. We run two spectra, one with REDOR, without REDOR, and one with REDOR. And if the two atoms are close to each other, we will observe some dephasing in the NMR spectrum. And we can clearly see here, when we do the detection on carbon, we have a decrease in the signal in the alanine here. Or when we do the detection on the N15, we can clearly see that we have a decrease in the signal, meaning that in the lipid bilayer, we are capable, when the peptides are together, they become close to each other. I will skip this part. Yes, then to finish, we are also developing another study in the team represented by a metabolomic study on living bacteria. The idea is to follow up what's happening to the bacteria when it's treated with antibiotics or antimicrobial peptide. And also NMR, solid-state NMR is a powerful tool to do such type of studies on a living bacterial cell. We know that we have different type of metabolites, and once the bacteria is treated with the AMP, what's happening? Does the AMP interact with DNA, protein, or metabolite? What is the metabolic change in bacteria upon treated with these molecules? And this will allow us to understand the biological behavior of the bacteria, and also will help us to develop also more drugs in the context of antimicrobial resistance. So the idea is to develop a metabolite library, meaning we have to acquire NMR spectra of the different known metabolite in bacteria. We have acquired for now more than 100, more 100 metabolite. Then we acquire NMR spectrum of the bacteria itself. It's a very complex spectrum. This is why we use some software for the data analysis to do the quantification or also the assignment of the data. And one of our preliminary results that I show on the acetate, in fact, we are capable to follow the release of acetic acid with time. And it's known that according to the literature that acetate is produced in anaerobic fermentation of the bacteria or also produced as a part of the degradation of the sugars of the cell wall. And we have done the experiment also in presence of the peptide, and we can clearly see that the release of the acetic acid is stable with time when treated with antimicrobial peptide compared to the control. So this is like a first proof of concept that this metabolic project is on the right way. And we hope that in the future we will really get more insight about other about what's happening into the in-cell compartment of bacteria. And with this, I finish my talk. I would like to thank you for your attention. And I would like also to thank my team, the Membrane Biophysics and NMR team, especially my supervisor, Buka Beschinger, Jesus for the NMR, Chris for the biophysical studies, and all the other team members. This is our NMR platform in Strasbourg, and in Strasbourg we have a very beautiful cathedral, the Notre Dame Cathedral. It's a very old one, and if you had the chance to visit Strasbourg one day, I really recommend you to go there and visit it. I would also to sincerely thank Jarbas, Rodrigo, and Amanda for all their help and hospitality here in Brazil, and Coffee Cup and Campus France who made also this my journey here wonderful. And thank you for your attention.
So thank you, Ahmad. We have time for questions.
Thank you for the great talk, Ahmad. I have two questions. One is regarding the method of your NMR. Is it possible to analyze larger molecules, like big proteins forming protein channels in this membrane?
Indeed. If we look to the literature, you will have a lot of examples of big membrane proteins. In fact, the advantage of using solid-state NMR is you can go beyond the limitation of the molecular weight of your protein. So there are some that, like the influenza channel, was investigated in bilayer structure. Even we have shown some A-beta peptide, amyloid peptide. So we have also a lot of examples of fibrillar assembly of amyloid peptide, like EAT-S, tau protein, and the structure of this peptide was well documented in the literature. So yeah.
But not just the structure of the protein, but the insertion, the formation of channels in the membrane?
Indeed, yes. We can follow the insertion and the interaction with the membrane and the folding of the peptide.
Oh, that's great. And regarding the metabolomics by NMR, is there a limitation of some classes of metabolites that are more favorable to be analyzed by NMR or by LCMS?
I think what we are trying to do in comparison to LCMS, for example, is to keep the bacteria alive. So we don't want to do extract. We do the bacterial culture, then we recuperate the bacteria, and we acquire the NMR data directly on the bacteria. So there's no extract. I think one of the drawback or one of the limitation is the solubility of some metabolites. Because when we acquire the metabolite library, we try to acquire the data in the same condition that we use for bacteria. So we can compare the data. Sometimes during the preparation of this library, you find out that some metabolites are not really soluble in PBS buffer, for example. So we need to find other solution for this. So yeah, one of the limitation maybe is the solubility of some metabolite, and yeah.
Thank you.
Thank you. Nice talk. Very difficult subject. Do you know if there are some difference between the metabolomic aspect from the normal bacteria and the resistant bacteria?
In fact, this is the idea behind this project. We are starting it now, but the future application is to take resistant bacteria, unresistant bacteria, and to study the metabolomic behavior of the two cells. So we get also more insight what's happening and what's going on. So yeah.
So there are no studies yet about it?
No, we just, it's just like we started this project.
Thank you, very interesting.
May I ask a general question? For decades, if you read reviews about pore formations of antimicrobial peptides in the membrane, you always see these explanations of the carpet-like model, the toroidal model, etc.
Right.
What is your opinion? Are all these different models still supported by novel data, or do you prefer one particular mechanism of action?
For me, I will go to my to Burkhardt model, the SMART model. In fact, as I mentioned, it's an interplay between lipid and peptide. Like, for example, maybe at low peptide to lipid ratio, the peptide will partition into the membrane in a carpet-like. If you increase the peptide to lipid concentration, maybe the peptide, they start to find each other, and maybe they can insert or create channel. The lipid composition also could have an impact on this carpet model or toroidal model, as we clearly seen in the N15 NMR data. For example, PGLA is transmembrane in saturated chain and is like a carpet model in unsaturated lipid chain. So yeah, it's really depending on the lipid composition on the lipid composition of the membrane, the charges, the fluidity, so, and also the peptide, because we can find some cationic peptide, very hydrophilic. We can find some hydrophobic peptide that they can really insert into the lipid bilayer. So yeah, this is really depending on the two factors, peptide and lipids.
And from my own experience, whatever the sources of the peptide from any species and venoms, there seems to be quite often a critical concentration. In my experience, it's 1 micromolar from a scorpion peptide or whatever. And then if you go higher up than 1 micromolar, you often see in a bioassay that we use, like electrophysiology, you see then pore formation. But is this also or is there an explanation why perhaps 1 micromolar for all these different types of peptides is a kind of critical solution that they indeed find themselves and then can go agglomerate and then form pores? Or is it just an empirical observation, this 1 micromolar cutoff?
I would maybe imagine like a CMC concentration, like beyond a concentration, molecule can start to find each other. Maybe they can create a larger assembly, and with this larger assembly, they are really capable to form these larger pores and create this activity. So yeah.
Thank you.
Is there any other question?
Hi, Dr. Saad. Thank you for a great talk. It's a very advanced level research. I have basic questions related to NMR.
Okay.
Like if we combine peptide with polymer, so is there any restriction for the NMR if we combine the peptide with polymer or other peptide, we will be still able to do NMR?
It's depending on what context you want to do this NMR. For example, if you want to study peptide polymer, for example, interaction in a solution, yeah, you can do some titration, check some chemical shift, and you can do this in liquid NMR. If it is in a more complex environment that you want to see maybe this in a membrane environment like lipid bilayer, it's possible, but here you also need to play with some factor like labeling, like we did with PGLA and magainin, to playing with labeling so we can distinguish what we are observing on our NMR spectra. So yeah, it's possible in the limit of what kind of information you are looking for.
Thank you. One another question. You explained the peptide has more impact on negatively charged membranes, right? So peptide has positive charge.
Yeah, they are cationic.
So any of the reason if they reduce or lose this positive charge, they will still able to show antimicrobial activity?
I guess if we look to the literature, especially for the magainin peptide, yes, if you remove maybe the positively charged residue, the interaction will be weakened. Because for me, this negatively charged lipid are the driving force to take this peptide and insert them into the lipid bilayer. So the interaction will be weaker in with their ionic membrane compared to a charged membrane. So with the charged peptide compared to uncharged peptide. So yeah, that really could impact the interaction with the lipid bilayer.
Yeah, so maybe interaction is loose, but if any of the reason they interacted, still antimicrobial activity will be normal, like they will be easily kill the bacteria, right? Just interaction will be reduced.
I don't have an answer because I don't have an answer for this question because I really don't have information about it. But logically for me, yes, when you if these are known to be cationic and the electrostatic interaction is very important to the interaction, and you are removing this, so you are weakening your interaction. So the activity will be lost logically.
Okay. Thank you so much. Thank you so
Welcome.
much again.
Any other question? So thank you so much, Ahmad, for the fantastic talk.